Good afternoon, everybody. I'm going to be talking about reusable devices for cleaning validations, what should be considered, and what you want to know about cleaning validations and how to be performed. My name is Alpa Patel. I work at Nelson Laboratories. I've been doing these validations for 14 years, so if there's any questions you guys have at the end, please do ask, and I should be able to answer anything regarding this topic. When we talk about cleaning validations, we're talking about anything that's reusable, which I'm going to be talking about today. It could be from surgical trays, from hip or knee. It could be endoscopes. It could be blood pressure cuffs. It could be monitoring systems. I know that we've done validations on keyboards. We've done it on laptops. We've done it on screens. We've done on ultrasound consoles. Things like that are also applicable. Anything that's reusable in the OR room would be classified as a reusable, and we would have to perform validations for that. A lot of the time when we talk about reprocessing, thorough cleaning, which includes the cleaning validation, is often misunderstood. And the reason for this is because these validations seem to be grouped together with sterilization or disinfection, and usually people think it's a decontamination validation where we're grouping everything together as it's proposed or as it happens in the healthcare facility. When you go to the healthcare facility, you start with cleaning or disinfection and sterilization. There it's understood that it's done as one process. But if you do validations, we're doing it in all different steps. And depending on how your devices are used is how they would be tested. For example, if your devices are thermal liable, then you can't do sterilization, and you would have to do disinfection validations. For example, let's say we have the slide in the OR room. We can't sterilize this, so we would have to do a disinfection validation. It's not, it doesn't come in contact with blood or anything. It's just sitting there, so we would consider it as a low-level disinfection validation. This is an example of a surgical suction tube that had debris in there that people thought a long time ago that if you skip cleaning and just do sterilization or disinfection, we're still fine because we're killing the bacteria. That is what causes infection. So why are we so worried about this cleaning aspect of it? But the reality is, if there's any debris, guess what? It's not getting cleaned and it's not getting sterilized or disinfected. And then the debris will be impacted over and over and over again, causing bacteria to hide in those areas, and you can't sterilize or disinfect them, and that's where the outbreaks happen. When you think about cleaning in your areas, the first thing you want to think about is what affects this cleaning. Device design, human factors, instructions for use, device compatibility, all these things play a major role in performing a cleaning validation. Often what happens is we come at the end of the process where, you know what, I have a checkbox and I have to go through by compatibilities first. Then I have to make sure that I've got packaging there and I've got bioburn tested, and cleaning and sterilization validations usually follow at the end of the process when you think about device validations or even design phase of it. These are some of the steps that I have outlined that if you think of cleaning validation, or if you're thinking of a reusable medical device at your facility, or you are manufacturing any of those, these are the steps that you want to consider. Design phase should be the first. When you think about cleaning validations, that should be the first thing that you want to consider, because when you don't consider the device cannot be sterilized or cleaned. Many times they are There are disinfectants that can discolor your device, or lubricants may not adhere to it and break them apart. So it's very important to ensure that these materials can handle the reprocessing supplies for it. Instruction for use, always have an instruction for use. If you don't, there are other predicate devices that you can use to see how to make an instruction. If you don't, there's always a third-party laboratory that can help you establish those instructions. Human factor considerations, these are big, of course. You have to establish human factors. Who is using these devices, and who is cleaning and disinfecting or even sterilizing these devices. And then development of a protocol, execution of protocol, and ready to launch. But all these steps have to be met for just a simple cleaning validation. So I'm going to go over every different areas of the process when you have to do a validation. We're going to start with device design. Cleaning validation should be considered during the initial phases. Often we see that it comes at the end, like I said, but always test on a prototype. If you have a device design that you want to see if it's going to work well for cleaning validations, is it going to get the debris? Is it going to be hard to clean? Do an R&D validation. What this entails is we test one device and we have one positive control. And we can tell you if this was a good prototype or it tells you something in a benchmark level where you have to what you have to work on and what you don't have to work on. We often see prototypes validated first, and then they will move on to the final design of that product. Evaluation of repeated use of devices with disinfectants and detergents. As you know, it's reusable, so these devices will be used multiple times. It might be 10, it might be 15, it might be 100, it could even be 500. We have validated a device that has been reprocessed 500 times. And you want to see the history that will it withstand these many times, or will it start to break down? At what point does the device start to break down? A lot of the times, the validations are performed with neutral pH detergents, but as you know, Europe, for reusable medical devices, use alkaline. And when they use alkaline, it breaks the devices apart more than it would be for neutral pH. So things like that also should be considered for reprocessing. Also, questions regarding biocompatibility on material breakdown has been proposed by the FDA to many manufacturers, that how do you know that after 50 times the material does not affect the patient? And this is something we're seeing recently being asked by the FDA, so we do repeat cycles and then do biocompatibility testing at the end of those 50 cycles or after the 10 cycles, depending on the use of your product. These are some of the difficult design features for you that should be avoided, but I understand in these days we can't avoid most of them anyway. But these are difficult design features for cleaning validations. When we see these features, when we see braided cables, the 3 braided cables are one of the hardest to clean, and once debris gets in there, it takes a lot to take it out. Same with dead end lumens. Once we inoculate those dead end lumens, if there's no flush port, it's stuck in there. It's like that suction tube picture that I showed in the beginning of it. Now, always think about flow, where there's a flush port and there's a flow through for liquid to go out. And inaccessible cracks and crevices, that's every device out there. They all will have it. Just have to make sure that we can get to them. Disassembly and reassembly instructions, very important. Devices that are intact, that have these kind of features, are very difficult to clean, and debris can harbor in those areas and stay there. I know that the FDA is pushing manufacturers to find a way to make sure that the devices can be disassembled at some point, that it's easier for reprocessing for the people that are are doing these. This is something that I think everybody should be familiar with. It's TR 55. It talks about human factors. Right now in the reprocessing industry, human factors is a big topic, and it's getting a lot of headway. The FDA is also looking into it very, very thoroughly and making sure that manufacturers adhere to this TIR. It was published in 2014, and so if you haven't looked at it yet, I strongly recommend that you do. One of the things that it highlights is it gives you instructions on how the instructions for you should be laid out. What font size you should use, how it should be presented, and number of steps. Avoid bullet points. Put them in steps. Put them in steps that are going to be performed. Step 1, you do this. Step 2 comes the cleaning. Step 3 comes disinfection. Step 4, sterilization. So in order of the reprocessing is what they're looking at. Also short and concise instructions. I think it's the next one. Yes. When you present an IFU with verbiage like this instead of a chart, I mean, what is easier to see? It's the chart. And especially the central processing people that are actually cleaning, the chart makes it much easier to pick out what you're trying to say in words. So if it can be in a chart form, that is more encouraged by the FDA than in a written way. This is something that I have seen in the past few years, is that yes, we do have instructions for use, and they're laid out really well. Everything is step by step. Everything is written as specified in the TIR, as specified by the FDA. But once it gets to the healthcare facility, what happens after that? Where is your IFU stored? Where is that IFU? Is it with the central processing unit, or is that IFU stored in another place? So what I have seen recently is that these IFUs have a condensed version of them, which the healthcare facilities make, and these they're very short. Like you can see here, soak the devices in detergent, while the first step has fully immersed the test articles into the prepared detergent and allow the test articles to soak for 5 minutes. So very short, very concise. Of course, in your IFU, you have to write it step by step, but this is what the healthcare facilities are doing at this point. It's in a binder. There's probably 8 cleaning instructions in there, and they're using those instructions to clean the medical devices. Some of the information that should be in an IFU are listed out here. Now, how specific do you have to be? I had a call from one of our clients saying the healthcare facility won't process our device in an automated washer because we have specified to use deionized water as the final rinse. And they don't have deionized water. So our response to that was that in your IFU, do write what you want. You don't have to write everything that you have validated to, but if you put treated water, which the standards allow, because the final rinse should be performed with treated water, then it could be any water that has been treated. And if it's going through a 0.2 micron filter, it's considered treated water. So very important to know those kind of aspects is how specific do you have to be in your IFU? Because the healthcare facility will call you and say, we can't meet this because of this reasons. And he had to delay his testing, and they had to pull it out. Same thing with the dental instrument. The client called us and said, well, we just wipe it with IPA. And they won't do it because it's not a cleaning. IPA is considered disinfection. So you don't have a cleaning process. So you have to validate the cleaning separately then the disinfection. So these are questions I just got this year. So it's not something that happened years ago. This is recent that we're still seeing. The reason why human factors play such a major role in the industry is because they are the people that are actually cleaning these devices. And if you can't make it easier for them to read and establish what you have put in the IFU, they can't perform their duty. And these people are usually with high school diploma. They don't have a lot of education, and they're looking at easy, simple language to follow. So very important that we do follow those instructions. When you're designing for validation, you want to consider environmental factors, PPE that's used in the central processing that the validation unit will wear as well. Supplies and equipment used should be available in healthcare facilities. A lot of times they specify you need to use this brush. If you can't provide it with your device, then you have to provide information where they can purchase it and where they can buy that brush from. And all the reprocessing instructions has to be completed from start to finish. You can't pick a certain portion of the reprocessing instruction and say just validate this portion, and that should suffice everything else. The entirety has to be validated. One thing that the FDA is harping on is manufacturers to go to healthcare facilities and do clinical validation tests by giving them the IFU and the product and say, here you go. Let me see if you can clean this easily. And they will be able to tell you if your IFU was successful or not. We know exactly how, when you come to a third-party laboratory, we are used to seeing these IFUs all the time. So we can read them really fast, but they do it every day, but it's not to follow every single step. So very important is that if you are considering doing a reusable, take it to the healthcare facility, have them perform it, see if they can even perform it to the step, and ask questions to the testers. And this is outlined in TR 55. Then an FDA, I can see in 3 or 4 years, they will mandate this for sure from the manufacturers. There was a study done in 2008 and 2009, I believe. It was a CLANR study, and 183 scopes were evaluated in a couple of facilities, and only half of them were reprocessed properly manually, and only 1% of all the steps were completed. And this is scary. I mean, we're talking about scopes, so yes, they have long instructions. They are pages and pages and pages long, but this does happen every day, and these numbers haven't changed. They're still the same. I mean, it needs time to improve, so hopefully in another few years we'll see an improvement on there. This is one of the FDA guidance document that if you're interested in reprocessing or are doing reusable medical devices, very important document to be familiar with. All the validation plans, what they need in the validation, what should be validated are all in this document. And they specify very clearly what kind of controls they want in place for the validations as well. Now I'm just going to go over some of the concepts of a cleaning validation from the document that I just showed you. I think it was about 5 years ago when the FDA came to us and said validations on newly manufactured devices will no longer be acceptable. You would have to perform validations on used condition devices because these are reusable and they do get reused over and over again, so we want to see them in used condition. Now, when you're manufacturing a new device, you don't have them in used condition unless if you've used them for clinical trials, and you can use those devices and provide a justification to the FDA. If not, then we do perform a test called simulated use cycle, where we're simulating the use of the life of a device and then test it at the end. Usually 6 cycles is what is recommended by the FDA. If you're going to ask me where the 6 comes from, we don't know. It's not written anywhere. It's a guidance that the FDA has given to manufacturers, and that's how we have come to know that 6 cycles is acceptable for these validations. Now, when we do a use, it has to be contaminated as it would be for the validation. It would have to be cleaned manually or automated, and either you go through disinfection or you go through sterilization. One or the other has to happen, and that is a full life cycle of the device and how it would be reprocessed at the healthcare facility. When you do these validations, you always want to consider worst case devices and worst case testing. What I mean by that is you want to use clinically relevant soils that are considered worst case. You also want to test locations that are worst case, dwell times that are worst case, which I'll go in detail as we go further. And in the cleaning side of it, you want to use minimum times, temperatures, and procedures as well. When you select a test soil for cleaning validations, these are some of the test soils that are out there to be used. Of course, the blood-based test soil is mostly and widely used around the world for validations. Mucus-based soils are there, but usually blood-based soils are used. A lot of the time, we do mixtures and additions to test soils, and the question mark is there to address that. I remember we had one validation we had to do for a liposuction, and it was a canister that was reused, and we had to come up with a soil to simulate the fat in the canister. And what we did is we took blood, added Crisco to it, blended it together, and it was exactly the consistency of the fat that would be removed during the procedure. And then we added it to the canister and tested it that way. So they are options that are open to mimic exactly what your device or your product would see during contamination. If you're not sure what type of test soil to use, there are standards out there. ASTM F3208 just got published this year, and it goes through many test soils in there and what devices are applicable for those test soils. There is also a working document in ASTM that we're working on, which talks about application of test soils, how to apply them on the medical device for validations as well. These are some of the contamination methods that I use for clinical validations. Either you can do a full immersion, you can do spray, you can also do handling with soiled gloves, or direct inoculation, where we just use a syringe to directly inoculate the crevices or areas that are very hard to clean. A lot of the time, they are mixtures that we would use where we would handle the handle with gloved soils, but the distal end of the device would be immersed in the fluid. When we talk about worst case dwell times, we're also mimicking that in the validation. We're trying to simulate the time it takes between procedure and reprocessing. What I mean by that is when you go to the healthcare facility, once it is not reprocessed right away. It takes time to go to the central processing unit where it's going to be processed, and then from there, it gets cleaned. It could be 8 hours, it could be 4 hours, it could be 72 hours before your device gets reprocessed. And that's what we're mimicking at this point. This is a real picture of a healthcare facility where the devices were waiting for cleaning. And you can see that even if the surgical trays are not contaminated from the dripping of the blood that are inside, the tray will go in onto the tray. So this is a new thing that the FDA is starting to look at, is cleaning validation on surgical trays itself. Not the devices that are in there, but the tray itself as well. Also, when you do validation parameters, how we evaluate is if you have an IFU, the validation is performed with less time, less soak time, less brush time, less flush time. So it will be less than what the IFU specifies for reprocessing. Selecting test sites. When the time comes for inoculation, now you've already figured out the test soil you're going to use. Now we have to figure out the sites. And a lot of the sites are determined on what portion of the device would be contaminated. And usually it is not just one device is what we test. There are certain sites, especially if it's large, we will test certain parts of the device. These are some of the easy ways of figuring it out. But for example, if you have a keyboard, some of the keys that we would test would be the Enter key, the backspace key, the A key, and the keys that are commonly used. Detergents is another thing that is major for validations. A lot of the time, detergents that are marketed in Europe, we can't find in US. So it is very difficult to figure out what is the best detergent to use. We do help figure out by MSDSs what chemicals are in there, and then we would use that to figure out what is efficient and what is equivalent to what US uses. Another thing is that US does validations for neutral pH detergents is what's used for cleaning validations, but Europe uses alkaline detergents, and unfortunately what that means is you have to perform 2 different validations for the same product. It's just different detergent types that you've used. Methods of cleaning, there is a manual method, there is mechanical. Of course, mechanical has been taking over, and automated is the number one way of cleaning any reusable instruments. It used to be manual, but to take the human factor out of it, we're moving towards the automated method, and washer disinfectors are predominantly used at the hospital. Although all 3 are part of the cleaning aspect for all the reusable medical devices. If you go to the healthcare facility, you will see that manual goes in first. They will pre-rinse the devices. Then it will go in a sonicator if the product allows that, and then goes into the automated washer. So all 3 are usually used. However, they can be validated as one system. They have to be validated separately. Mechanical fits in the manual, but manual cleaning and automated cleaning are 2 separate things, and they have to be evaluated separately. Any cleaning supplies that has been used for the validation, usually the FDA will ask what kind of brush was used for the cleaning. If you use any pipe cleaners, what size did you use for those? And they are specific, and they do ask for specific reasons, because a lot of times when you're cleaning a lumen, if it's not a tight fitting, you're not cleaning anything. So the FDA wants to make sure that during the validation you use the right instrument, and also during your IFU, when you write it, the right brush size has been expressed in there. Some of the key things to remember for automated cleaning is you want to pre-wash all. You want to perform a pre-wash of all soil devices before placing in the washer, like manual rinsing under the tap water, very important. Once you place it into the washer, place the device in a way to facilitate drainage, and validate using high motor speed. We just had an incident last year where a sponsor was using low motor speed, and they were failing all of their automated cleaning because it was low motor. But once they pushed it to high motor, because that's what the healthcare facilities use, everything worked out really well. And always perform a drying step for the automated cleaning. That is these are the 4 success steps for any automated cleaning. If you do all these 4, you should be able to have passing, at least something that gives you an idea that I'm going in the right direction. Some of the acceptance criteria, for these validations, the FDA likes to see at least 2 fully quantifiable assays, which the guidance document goes in detail. Protein, hemoglobin, TOC are the top 3 that are recommended. Now, TIR30 has all these markers established in there. These are developed through a study that was performed on endoscopes, and since it's considered to be the worst case device in the industry for reprocessing, you can use that benchmark to leverage the other reusable medical devices. Another thing that we're seeing commonly asked by the FDA is cytotoxicity testing after cleaning validations to see if there's residual detergent left on those devices before it goes to the next step. And that is usually performed through a minimal essential media MEM test, which is a very simple test, very easy test, and it only requires one device for it. But keep in mind that that is something that we are seeing recurring over and over again by the FDA. The next steps for reprocessing or cleaning validations is try to coordinate the IFUs for similar devices. For example, there are 10 different IFUs for hemostats out there in the industry right now. Why do we have 10 different IFUs for one simple device like that? Can we just make it into one? Therefore, the AAMI group are working onto NX in TRI-12 to group these devices. Like you have for sterilization, you have a table that specifies, if you don't fit into this table, we can't help you. You have to fit into this table. That's the same concept they're trying to bring on the cleaning validations. If you don't fit into the 6 categories, you have to reconsider your product again. I mean, it's going to be a couple of years before we come to that, but we are working together to figure out how do we categorize these 6 categories and what should the instructions look like. Right now, they've moved from 6 to 4 categories, although there's still a lot of debate that we need to have 6 options available. These are some of the references that I have placed. I think there's 3 or 4 slides and also little webinars that you guys can look at for any references you need regarding reusable medical devices, cleaning, disinfection, or sterilization. If there's any questions, I can take them now. Yes?
Hi. Hi. Thank you so much for a very nice presentation. I have a question about your simulated use cycle slide. Mm-hmm. That was very interesting. I'd never heard of the 6 cycle recommendation. That's the first time I've heard of that, so thank you for that. I wanted to know, what is your experience with the order in which you perform the simulated use, whether it's acceptable to group all of your cleaning cycles together and then perform your disinfection cycles and then perform your sterilization, or if the FDA's expectation is to do the order, cleaning, disinfection, sterilization, then repeat again, cleaning, disinfection, sterilization. Did that make sense?
That does make sense, and that is a very good question. We get that question all the time. Yeah. What the FDA does recommend is one full cycle of the device, of how it will be reprocessed in the healthcare facility. Therefore, it would take on contamination, and then would have to clean and disinfect, and that's cycle 1. And then we move on to cycle 2, again, contamination, cleaning, disinfection, and then so it has to be performed that way. Unfortunately, yes. Any other questions? Oh, jeez.
I got a couple questions. The first one, you had the steps for worst case, like processing a tray with the pre-soak and that. Let's say that, from my experience, the reality is that they typically, a lot of hospitals skip some of the pre-soaks. Mm-hmm. They'll jump straight into the automatic washer. So wouldn't we want to validate worst case without the pre-soak to make sure that if we know that's industry practice?
That is correct. That a lot of times those steps are missed, but majority of the time that I have gone to healthcare facilities, there is a pre-wash in the way before it goes to either ultrasonic unit or in the automated washer. But you're right, small facilities do perform. If they don't have a pre-wash in there, they go straight to the automated washer. Now with that, when you have lumens and you have design features that need the pre-wash in there, you're just setting yourself up to failure at the end where the devices would have to be rerun twice or rerun 3 times. Therefore, we strongly recommend that you would want to perform at least a pre-wash and recommend the healthcare facility that they have to do the step before they place it in. Okay. For lumens and other devices, I know they have to do that, but simple devices, yes, I have seen where they have just placed it in the washer.
Okay. And then on, like cannulas, lumens, shavers and burrs specifically with valves inside, they make, the rack systems that are direct port and sprayed. Is that where you want to set your validation on a design rack system for that, knowing that maybe hospitals don't? Or would you want to take those devices and go more of a traditional open rack sprayer? Does that make sense? Like,
yes.
Do you want to make your validations fit to what's designed best for them? Or what would possibly be worst case that they see in the hospital?
We are setting our validation design on what's worst case that's happening at the healthcare facility. Majority of the healthcare facility don't have those racks available to attach to the lumens that can be flushed through easily. So that's where the pre-wash comes into play, and that's where. But we are, if recommended by the manufacturing facility, that they need that rack for the validation, they have to specify that in their IFU that you would have to purchase this rack, and this is the rack that you have to process our medical device. So we do have option to do that. But we always default on the worst case.
Okay. And I'm sorry, I have one more question. I know I'm totally hogging the mic. So it actually plays off of your question on repeat cycling. If you were able to show equivalency on running back-to-back sterilization and back-to-back automatic reprocessing and show that that's equivalent to doing the steps, have you seen that successful with the FDA, or are you forced to have to do the slower traditional or the...
No, that's a good question. We have had manufacturers to save time to do it that way because it's faster to do all cleaning 6 together and all sterilization 6 together. It's definitely faster, but we've had manufacturers do that route and put their 510K submission, and we've got a bounce back from the FDA going, this is not the way we want to perform the simulated use cycles. They have to be performed how they are being tested at how they're being reprocessed at the healthcare facility as a cycle, as a full cycle.
But if you could, has anyone tried to show equivalency? So
they have not.
Okay, so run 10 cycles in that order and show that it's the exact same as doing the other 10, and then leverage that to go up to 500?
I don't think anybody has done that yet.
Okay.
Any other questions or comments? Well, thank you, everyone, for sitting by and listening.